Good evening, everybody, and welcome to our latest hot health topic. It's my great pleasure to introduce Dr. Tania Slatter. Tania is a Senior Research Fellow and a Sir Charles Hurkis Fellow in the Department of Pathology, and she's involved in molecular pathology and looking at, in particular, outcomes for cancer. And tonight she's going to be talking on her work that she's done around brain cancer, so glioblastoma and others, and looking to see what we can see from a molecular point of view in terms of developing therapies for the disease. So thank you very much, Tania.
And thank you for the invitation to speak tonight. So I'm going to talk about the work that I've been involved in looking at trying to improve the outcome for brain tumours. And a lot of the work is done here at Otago, but we also include other centres in New Zealand as well. So this slide might be self-explanatory. So what I work on is brain tumours, so tumours that are in the brain. But for some of you, you'll be aware of this and some of you won't, that brain tumours can arise from cells that are within the brain themselves. So we call those primary brain tumours, and they could be malignant or they could be benign. And then we have another group of tumours that are aggressive tumours that start elsewhere and move to the brain. So these are metastatic tumours, and they're more common in the population. And the problem with brain tumours, as many of you can imagine, it's a very specialised organ. So a lot of the symptoms that patients will develop are those that would affect the functioning of the brain. So often patients will present with things such as issues with sight or hearing, or another major one is personality issues as well, which you wouldn't see with other types of tumours. The other part is we have this nomenclature for how we name brain tumours. So there's over 120 different types of primary brain tumours, and they have this specific nomenclature. So some of these terminologies can be quite confusing. And so the primary brain tumours are named after the cell of which they're thought to start from. So if you think about it, we've got tumours that are in the meninges, which are the coverings of the brains, and those tumours are called the meningioma. And the one I'll talk about today is in the main part of the brain itself. And these are gliomas, or cells that arise from glial cells. So glial cells are the cells that support the neurons in the brain. And the tumour type I talk about most is a tumour called glioblastoma. So it's a type of glial tumour. It's a very aggressive tumour, and it has other terminologies that people might be aware of as well. So glioblastoma multiforme, or GBM, or grade 4 astrocytoma. So it's a very aggressive tumour. The incidences of brain tumours aren't as high. It's only about 226 each year in New Zealand, but the mortality associated with them is very, very high. So it's a second leading cause of death for men aged 20 to 44, and it's also associated with a very poor overall prognosis. So the overall prognosis, we look at the median survival, which is only 14.6 months, and also the 2-year survival, which is about 2-year survival is at 24% of patients. So this is very low when you compare it to things like breast cancer or prostate cancer, where we tend to refer to as 10-year survival. So you can see that's a very aggressive tumour type. Unlike other types of tumours that you might be aware of, often there's really strong risk factors, so things like UV light for melanoma or smoking for lung cancer. But with the glioblastomas, we really don't have any strong risk factors. We can't identify who is most at risk for these type of tumours. So it's a very difficult tumour to try to identify who is at most risk. How we treat these tumours, so we've got Ahmad Taha here, he's a neurosurgeon here, so he will take that tumour out and try his very best to get as much of the tumour out as possible. And then we have radiotherapy that we use after that is to mop up all the residual tumour, because these are very invasive tumours. So the surgery is as hard as Ahmad tries to remove the tumour, there's going to be some residual tumour present. And then after radiotherapy, we treat with chemotherapy, and that drug is called temozolomide. So the brand name here is on the right-hand side, Temodol. So it was approved by the FDA in 1999, and it's following a protocol called the Stupp protocol. And I'll talk a little bit about that later. So that was the evidence-based medicine that temozolomide would work for patients with glioblastoma. And what we do in New Zealand is you get given temozolomide concurrently, so at the same time you're receiving radiotherapy, and you get it afterwards as well. So temozolomide, so some people will be aware of the difficulties in getting pharmac to approve drugs that work. Temozolomide, so you remember that Stupp protocol where they showed that temozolomide worked really well. It came in pretty quickly relative to other drugs in New Zealand. So 2005, that study was published. By 2006, pharmac funded temozolomide for glioblastoma patients in New Zealand, but there were problems. So some individuals, if you had a grade 3 tumour, so it's going to progress to grade 4, you were not given temozolomide. And in 2010, pharmac changed that decision. So this is the Dr. Moody here. There's the medical director of pharmac, and this is his response, saying that aggressive brain tumours are difficult to treat. We know that temozolomide works, and therefore this was the decision that they were going to include grade 3 patients into allowing them to have temozolomide. And again, as pharmac probably say with many drugs, saying, well, yes, these drugs work, but they're expensive. And pharmac have to try their best to make sure that they fund things that based on evidence-based medicine. The next decision was in 2016 because there were still issues with the funding of temozolomide. So if you were a patient and you were living for 2 years with glioblastoma, so temozolomide is probably working for you, you were not eligible to anymore. Once you finished your 6 adjuvant cycles, that was it. And that seemed very unfair because we should be in a position where if drugs are working for patients, that they're able to continue on those drugs. So in 2016, pharmac decided that if you showed evidence that temozolomide was working for you, you were eligible for more temozolomide. So that's to show that that was done as well. And this is the evidence here of why we use temozolomide. So in this case here, this is the Stupp protocol published in the New England Journal of Medicine, where we all want to eventually get a paper in. And it was in 2005, and it showed that, and if you look at the red line, these are patients. This is patient survival, and it's showing that they survive for a shorter period of time if they're just given surgery and radiotherapy compared to patients that were also had temozolomide. So this was the evidence that temozolomide improved the median survival. So that's where 50% of the patients are surviving. So originally that's why it's now 14.6 months, and longer term survival. So that's patients that are surviving, if you take 2-year mark, we've got more people surviving at 2 years, about 20% to what we had before temozolomide was available. So temozolomide is improving the overall prognosis for 50% of patients, and also improving the longer term survival for a group of patients. So the previous with temozolomide, that's drugs that target the tumour cells, the cancerous cells. But we have another issue in trying to target tumours effectively, is that tumours have other cells that are present. So it's not just the cancerous cells or the malignant cells. A tumour contains many other cell types. So these can be immune cells, and these immune cells can contribute to that tumour being more aggressive. And you can also have other cells, such as endothelial cells, that produce new blood vessels. So that's a problem because those brain tumours will eventually run out of nutrients, and those new blood vessels will deliver more nutrients to those tumours. So if you want effective treatments for people, it's not only a case of targeting the malignant cells, we also need to target these other cells that promote this tumour microenvironment. And some people might be aware of these. These are particularly big drugs that came in for treating patients with melanoma. So we have checkpoints in tumours. So checkpoints are where the immune system, so the immune system is kind of got two different arms, I guess. You could see it as evil or not. So the immune system is very good at getting rid of tumours. The immune system can target things that it considers to be abnormal, such as tumours. And that's a function of the T cell. But unfortunately, the immune cells also have a function in which case they can stop that T cell from being effective. So it can stop those T cells from killing your tumor cells. And they do this through checkpoints. So one checkpoint is a protein called PD-L1. So you can have PD-L1 on your tumor cell or another cell in that tumor microenvironment. And that inhibits by binding to something called PD-1 on your T cell. So that combination of those two molecular markers will stop that T cell, so stop the immune system from being able to target your tumor. And we have other ones as well. So it's not only the PD-L1, there's something called CSF1R. So CSF1R is another immune cell that's present in those tumors that stops your immune response from being able to target the tumor cells. So again, what we want to do is in the new therapies that are coming out are not just targeting your malignant cells, they're also targeting that immune suppressive tumor microenvironment. And this is just to give you an indication. There's many, many different drug trials at the moment that are using these immune checkpoints in patients with glioblastoma. But there's a particular problem for that. And that is that at the moment, how we design drug trials is we target all patients. Regardless of what your outcome's going to be, everybody gets the drug target. And you can see in this slide here, this is outcome from patients in Hamilton and Dunedin, that if this is post-temozolomide era, the outcome is very, very different for individual people. So some individuals are surviving a very long period of time, 40 months, and others are having a very, very short survival time post-diagnosis. So this is a problem. So the outcome for this type of tumor is very, very heterogeneous. It's different for different individuals. And the reason for this is that the tumors are not the same. We call them glioblastoma, but there's lots of differences within those tumors and between individuals. So the key thing now is we need to identify what those differences are. Why does one person get a really good response to their treatment while another person does not? And target the individuals or people with all our tumor subgroups much, much better. So we can improve the outcome for all with glioblastoma, not just a subset of patients. And the reason why those tumors are different is because they have different genetic changes. So in your tumor, some people might have one genetic change, another group will have a different genetic change, and that will affect the response to treatment. And so genetic changes affect the way cells grow, they affect the way cells die, they also affect the way cells will respond to treatment, and they'll affect the way that they can evade detection by the immune system and destruction by the immune system. So for example, in my previous cases, your tumor might have PD-L1. So you're going to have an environment that's more likely to suppress the T cells from working versus a patient who doesn't have PD-L1. And again, these genetic changes contribute to lots of different things with the immune system. Lots of different things to the tumor. They contribute to the development of the tumor, the progression of the tumor, and the resistance to treatment. So this is a slide of how we work at Otago. What we do is we have a system where we start with understanding what's important for patients that have glioblastoma. So this is where we get patient samples from surgery, and we look at, in those patient samples, what are the key genetic changes that are in those tumors that will predict prognosis or treatment response in our patients. We then move to more lab-based things. That's the cell culture hood. It's very well organized cell culturing conditions here. And so what we want to show is, although we found something important in a tumor, does it really cause the things that we think it does? So what we want to do in that case there, we can engineer cells to produce the thing we think is very important and prove that it actually does promote tumor growth. And so it's kind of that it is not just associated in tumor for some other factor, it is actually causing that resistance to treatment. And the last part is that we can use animal models. So these are key. Once we find something in patients that's important, we then translate that in vitro to show, yes, it does do what we think it will do. And our animal models can actually prove that, yes, that thing that you thought was promoting that tumor actually does. And I'll talk about some of the work that we've been doing in relation to glioblastoma. So this glioblastoma has these key genetic changes. So some patients will have some mutations, and these are called by these long names. I've just given the shortened version here, such as IDH1 mutations. Some patients will have P53 mutations, and some will have different means by which cells become immortal. So these are things at the end of the chromosomes that make those cells able to continue dividing or continue surviving. And there's different mechanisms for that. So one is, we call it ALT, and one we call telomerase positive. So different patients will use different mechanisms. Another a marker in glioblastoma is a genetic marker called MGMT. So MGMT is key, so some patients will have MGMT in their tumour and some will not. If you have MGMT, you're able to detoxify temozolomide. So temozolomide will damage the DNA of your tumour cells and cause those tumour cells to die. But unfortunately, if you have MGMT, MGMT takes off that DNA damage, so your cells are able to continue surviving. So it's a marker that means that you're likely to be resistant to temozolomide. Another marker that we've been heavily involved in is some patients have tumours with lots of those cells that promote a tumour in the tumour microenvironment called macrophages. So some patients will have on the left-hand side, they have a very low content of macrophages, and others have a very, very high content. So you can see the image on the right-hand side has a lot of these macrophages in this particular patient's tumour. So it's very different to another patient. And what we've been doing is, and I showed you that slide before where I tried to illustrate that GBM is very heterogeneous, patients survive for very different periods of time. But what we've got here is if we're able to use our molecular markers, we can start to better stratify patients. So in pink at the top, we have a tumour that uses one cell immortalization pathway. It has IDH1 and P53 mutations, and these individuals have a better prognosis compared to some of the other tumour subgroups. The group in green in the middle, this is an intermediate prognosis. So these patients use a different cell immortalization pathway called telomerase. They have a low content of those tumour-associated macrophages. They don't have IDH1 or P53 mutations. And the group in blue at the bottom, these tumours are very similar in some respects to the group that's in green, but they have a high content of tumour-associated macrophages. So they have a much poorer prognosis. And the other key thing is we can then look at data. The previous data I showed you was after temozolomide was made available in New Zealand, and we can compare how things have changed now that temozolomide is present. And so that's the tumour group on the right-hand side. So what we've been able to show that in that group where they had the better prognosis before, they had a better prognosis before temozolomide was made available. The group in green is key when it comes to understanding why some patients survive a lot better with temozolomide. This group is the group that's responding to temozolomide. So before temozolomide was made available in New Zealand, your 50% of patients would live 9 months and 0% of individuals would live out to 2 years. Now that temozolomide is made available, we've improved that by 18 months, which is a long time for patients with glioblastoma. And 20% live more than 2 years. So if we look back at the previous Stupp protocol, where they also found 20% of patients living out 2 years with temozolomide, we're able to show this is a subset of glioblastoma patients. They have a particular genetic changes in their tumours, and they're the ones that are getting the benefit from temozolomide. The group in blue, they had basically poor prognosis now, they had poor prognosis before. So they're not getting the same benefits from temozolomide that the group in green are getting. So what we really aim to do with our research is improve the prognosis for everybody. So particularly what I'll talk about now is how we're working on that group in blue, the ones that have a very poor survival that was very similar to the group in green before temozolomide was made available. So maybe we could find a drug that would target this group. And this is just that's what we're trying to say. So is there a molecular difference in these tumours? So can we find the reason why these tumours are resistant to temozolomide and why these individuals have a poor prognosis? So here we come to P53. So Antony Braithwaite's here, and I started working on P53. So P53 is, for many of us, it's very familiar, and other people wouldn't have heard of P53. So P53 is a gene, and its normal role is to make sure that the genome stays intact. So when you have a cell that gets targeted by damage, P53's role is to stop that cell from continuing, so that genetic change is not transferred on, so that those cells stop proliferating or they die. The problem that happens in cancers is that you mutate your P53. So you have mutant P53, so your cells, when damage occurs and you start to get genetic changes, you don't stop those cells from dividing and you don't kill those cells. So those cells will continue on to develop tumours. And that is the reason why many human tumours have P53 mutations. Unfortunately, that doesn't explain our group that has a poor prognosis. They don't have P53 mutations. But recent work has shown that P53 mutations don't explain everything about why affecting the P53 function causes tumour. There's things that are very, very similar to P53 mutations. They are called P53 isoforms, and they also promote tumour development. So they're very similar to P53 mutations. And what they are is they're missing the first part of P53, so they have an N-terminal deletion, and they have subsequent changes at the C-terminus of P53 that leads to very different isoforms. So I'll focus a bit more in the later part of the talk on the delta 133 P53 isoforms. So again, the key thing to remember is, like mutant P53, this is a different form of P53 that's just a very emerging field, and they also promote tumour development. So this is work that we've done, and it gave us the evidence that this could be key. If we look at glioblastoma patients, and we see we've got some that have mutant P53, but in a group that didn't have P53 mutations, they had high levels of these P53 isoforms. So that led us to believe this could be key here, because if you imagine tumours as they promote, they need to have mechanisms that are going to allow them to survive, and you can do that by mutant P53, but you could also do that by increasing these P53 isoforms. And that's what we've gone on to show in the slide that's on the right-hand side. So if you have P53 isoforms, they're increased in our group that has the poor prognosis. That's what we would expect if we're trying to say that maybe these isoforms are associated with more aggressive tumours. So we've got evidence using human patients that would be the case. We've gone on to show that is the case. If we want to show that this marker is causing a poor prognosis in patients, we need to show that this marker is in the tumour cells. And that's what we've done here. So on the bottom panel, you see some black arrows. That's pointing out where that P53 isoform is in the malignant cells in a glioblastoma. So that's what we want to have that evidence to suggest we've found something that's increased in tumours, but it's actually something that's found in those malignant cells. So that's consistent with those cells being able to cause problems, such as preventing those cells from dying or preventing those cells from responding to temozolomide. And the other piece of information that we've found is that P53 isoforms become increased in tumours where the hypoxia is developing. So hypoxia in a tumour is where you're running out of the blood supply, and usually that would signal many cells to die. But tumours develop mechanisms to survive in those conditions, and that's what's happened here. So we're finding that this isoform is increased in the malignant cells in glioblastoma. We also find that in these tumours that have a high content of P53 isoforms, they have high content of those things that affect the immune checkpoints. So high levels of CSF1R and high levels of PD-L1. So this is adding to the fact that these tumours would it be preventing the immune system from targeting those tumours. But it also means that maybe that these tumours could be targeted with these new checkpoint inhibitors. So these drugs that are preventing, that are inhibiting PD-L1 or inhibiting CSF1R might work for these specific patients. So in the next part of the talk, we've found this information in patient tumours. We've found a reason why that group that has a poor prognosis, why they may be having that poor prognosis, because they have an increase in p53 isoforms. So now we've actually done some work to actually prove that that may be the case using the cell in vitro techniques. So Marina, and she's here, Marina spent a lot of time engineering cells so we've got some cells that have the p53 isoform engineered in them. So we're able to look at, if you just change one thing in those cells, would we predict that those cells would form a worse tumour? And that is what we predict is the case. So if we engineer these cells and we only have the p53 isoform change, we get increased PD-L1. So that's consistent with the isoform leading to issues in human patients. We've also got another marker that we use. This is a CCL2 gene expression. So again, just another marker. If that marker is present, you would bring in more macrophages into a tumour. So it's a marker that drives in macrophages. So we have this increase when we just change that one, make that one genetic change. And this is work by Ramona, who's a PhD student in the lab. And she's shown if you just change that and look at temozolomide, what you can see is that the cells that have that one genetic change, if you treat them with temozolomide, there's no difference in cell growth. So the blue and red lines are exactly the same when you have that p53 isoform present. So the temozolomide is not able to stop those cells from growing. But in the control cells, so very, very similar with the exception that they don't have the p53 isoform, they are susceptible to temozolomide and other drugs as well. So with all of this work, so we've found something important in patient tumours. We've then looked at things in the lab and shown, yes, this thing would cause all these different markers that would be associated with a more aggressive tumour. The next bit of this project is to look at an animal model. And that's what's just being developed at the moment. So we're developing a new animal model that only has this p53 isoform change in the cells that cause glioblastoma in patients. So that's glial cells. So we're engineering them. So then we can look at, if we just make that change, will we develop glioblastoma? Will that glioblastoma have change the immune cells that are present? And will they be resistant to temozolomide? And if we establish this model, one of the biggest issues in the glioblastoma field is there's no good models for testing things. And that's hopefully we'll have a model that we can look at. And then we can test for new drugs. So if we use those immune checkpoint inhibitors, if we treat with PD-L1, will these patients have improved prognosis? And what combinations of treatments that we can use? So that's the future work that's going to occur. And just in the last bit of the talk, so I've talked about you look at something important in patients, you translate that to the lab and you make sure that it's important, then you put it into an animal model, which can again give you more evidence that this is something you should target and gives you evidence of what you could target it with. The next bit is constantly as we're doing this, we're also including clinical trials along the way. I've got two that I'll talk about. So I'm involved in 2 clinical trials at the moment. So this one here, this is basically using our way of subtyping patients before using all the molecular markers that we've found. And this is a prospective study done here at Otago and up north in Waikato. So what we're trying to do here is making sure that will that group in green that we had before that respond to temozolomide, will they get those patients get an even better prognosis now that temozolomide is available for longer periods of time? We suspect that will be the case. So that will give us more evidence that identifying this group of patients is really important to identify those individuals that will respond to temozolomide. It'll also give us more evidence that the other groups are not responding to temozolomide and we need new treatments for those individuals. And the second trial is very important. And Zhou is here from radiology, and she's been very, very key. We've done a lot of work looking at using radiology to better image our subgroups. But this is a new trial that's about to start in Dunedin. It's a world first. And what it is one of the properties of these macrophages is they can scavenge iron. And so what we can do is we can give patients a type of iron, a small particle iron, which is called USBIO. The macrophages will take up that iron, and we can actually image those macrophages in the tumour using MRI, because it's a type of MRI contrast agent. And that is very, very powerful, because not only will we be able to image a poorer prognosis before surgery, this contrast agent might be very, very key at getting a better image of that tumour. So we might get so Ahmad, for example, there's a neurosurgeon, might be able to remove more of the tumour because we've got a much better way of identifying the tumour. We also, so we envision that will be two main benefits to patients at Otago. We'll be able to predict worse prognosis before surgery, so the surgeons will be aware that this is going to be a very aggressive tumour. We might be able to get better image of that tumour to remove a much better part of that tumour. And in the future, the key thing is at the moment we're very limited at identifying people once their tumours recur. So once patients get given radiotherapy, the current way of using MRI doesn't work as effectively. And you get what we call pseudo-progression, where it looks like there's a tumour present, but it's not. And that's to do with issues with using the current contrast agent. But this agent might be a much better way of picking up tumours much earlier when they recur. And the other benefit is, of course, imaging macrophages is going to have a lot of benefits, being able to see if some of these targeted therapies are working. Because we can't go into, if you can imagine, if you've got a brain tumour, we can't just operate on you constantly to see if your tumours come back, but we can, if we were able to use MRI, we'd be able to image patients during a course of time and pick up that tumour much, much earlier. And we'll also have a means of being able to see if the treatments are effective. Jean's here tonight, so she can answer questions in relation to the tumour trial, but this is Jean's slide here. So this is what Jean's going to do. They're going to be, and Ahmad as well, they're going to be screening patients that come into Dunedin to identify those that have glioblastoma. They're going to be consenting patients to take part of this trial to see if they would like to have this very new MRI to better image their tumour. And then we're going to follow up and check that the macrophages do take up the iron, because we suspect that will be the case, and that will be very important knowledge if they do. And there's many, many people to acknowledge. So I've tried to acknowledge people along the way, just to make sure that I don't exclude anyone. So it's very important work, and it involves a big team of people having to work together from the clinical side of things right to the scientific side of things as well. So thank you.
Thank you very much, Tania. That was fascinating, and a great example of that real bench to bedside translational research, where you start with a patient and a clinical issue, you go back into the laboratory, you look at some of the key things that are going on in the laboratory, and then try to redesign some different changes to the way that therapy works. So do we have any questions? I guess I'll start. So is it just macrophages? Do you think it could be other cells, immune cells as well, that are involved? I mean, I know you've mentioned T cells, but could it be other granulocytic cells that are coming in that maybe express some macrophage markers, but they're not sort of true macrophages?
Yeah, so we suspect that, and hopefully we've had some work from your lab actually, Sarah, to help try to identify identify these immune cells in much greater detail using flow cytometry. So at the moment we're limited, we've done immunohistochemistry, but we don't actually know what these macrophages are, or if they are macrophages. And a lot of work in the literature says, they will be this, but we're proving more and more that the type of cells that are in tumours are quite heterogeneous, and they need to be identified with much greater precision than we've been doing.
Okay, we've got some.
You get enough material to use the cytoph, for example?
So if Ahmad's right here, so we ask Ahmad for as much tumour as possible for everything. So presumably we could. So that's a technique that we can use to look at protein peptides. To identify macrophages. They can use so many different immune cells. So that would be possible. Yeah.
Just a related question.
Yes. Yeah. So there is CD68 as well. So, but it didn't correlate as well as the CD163 is the strongest marker. So the other problem you might have seen, someone might have picked up there as well. The other issue in the literature is everybody says, oh, if they're CD163 positive, they'll be CSF1R positive. Why are you bothering? But they're not. Or that subgroup that have the isoform present do have more CSF1R positive cells, but there are tumours that have a lot of macrophages, CD163, that weren't CSF1R positive. So again, it's bringing home the message that what's happening in tumours is quite different to what you would predict if you don't look at patient samples.
Can you talk into the microphone, because we've got people watching online.
Okay. I'm just wondering if there are any interim results from the checkpoint inhibitor trials in glioma and how they're looking. Are they related to the expression of PD-L1 in the patients?
To my knowledge, people haven't looked, and a lot of them are failing. And as you can see with that group that I showed you, the problem is when you've got heterogeneous group of patients, you've got patients that will survive a long time regardless of what treatment you're giving them, so that group in pink. You have the group that are responding because the trials still have to give patients their current treatment, which is temozolomide. So you'll have groups that are responding to the temozolomide, and then you'll have groups that possibly would respond to the PD-L1 inhibitors, but if you don't identify them earlier and look at them before and after PD-L1, you won't identify them. So if you look at your cohort as a whole, you'll say, oh, the drug's just not working. So it's something that seems obvious to do, but people still aren't doing it, checking which patients you should be targeting first.
I think with the glioblastoma being heterogeneous, so there are many cell lines being there in the tumor itself. So whatever you take out with one drug, it's like bacteria, you know. You kill one type of bacteria, the other one will flourish. And then you have this competition. So you take the one with temozolomide and you leave the others which like flourish later on. This is why temozolomide fails after a while. Otherwise, it would kill all the tumor cells. And this is the problem. It's now what we're looking at, personalizing tumors. I mean, patients with different cell line, they should have different chemotherapy or different approach. Some of them surgery is better than radiation. Some of them surgery doesn't make any difference. So this is where this excellent work being done by the group. I'm really proud to be part of it because I think most of the work being done by these people. I mean, I'm only the technician. So they are the brain, I am the muscle. So,
I mean, I can just, obviously I'm an immunologist, so there are a lot of trials at the moment looking at combinations of checkpoint inhibitors for exactly that reason. If you just target one, you use one checkpoint inhibitor, you'll remove a certain population of cells or take off the checkpoint, but then others can come in, or and there's different kinetics of when they come in and out of a tumor. So there are combinations that are going on in a number of
and the tumor might be adapting. I mean, those cells are really live. They change. So whatever you do, they start resistance because the second time or third time revisions, they change with radiotherapy, with chemotherapy. So it's not going to be easy, but I think we are really on the right path, I think.
I've got one more question. So we've started looking at obese versus normal weight and looking at some of the changes we see in different cancers, not glioblastoma, but actually also looking at some of the changes that we see in immune function in obese versus normal weight. And one of the key things is macrophage infiltration and the way those macrophages work in people that are overweight. Have you ever looked at that in your patient cohort?
As far as I'm aware, most people are not overweight, are they?
This is an excellent idea because there was some talk about diabetes, glucose.
Yes.
So there might be association between this. I mean, with the obese patients, they're resistant to insulin. I mean, if they become diabetes or diabetic, I think Noelyn was talking about the increase, the poorer prognosis with people with diabetes. I mean, we looked at other parts of immuno. We have some patients who have, for example, asthma. we thought they do better than rheumatoid arthritis. They do better, for example, than patients who don't have that. Is it because of the increased expression of the P53 in asthma, for example? There's always, we looked at that. We looked at as well of interferon as part of possible treatment. As well, is just noticed from patients cohort that many patients who have glioblastoma, they will tell me they've never had a flu in their life for some reason. So this is where the interferon came to place, and this is what came about resistance and obesity, diabetes, other medical condition.
So metabolic dysregulation. Are there any final questions just before we close up? Great. Well, please join with me in thanking Tania one more time for a fascinating talk.